Hello and welcome everyone. In today's video, I will be teaching about the tryptophan operon, or we can say it as the trip operon. In the last video, we studied about the regulation of gene expression in prokaryotes by giving a special emphasis on the lac operon. Lac operon. In today's video, I will be explaining the regulation of gene expression by understanding the tryptophan operon. Okay, so my suggestion will be you first watch the part 1 of this video so that it will be easier for you all to understand this second part. So I will give the link in the description box as well as I will try to put it in the I button. Okay, so let's begin today's video. So tryptophan operon. Before going into the tryptophan operon, I'm giving a brief introduction what we learned about the operon. As we know, operon is of two types. What are the two types? One is inducible operon, and second one is repressible operon. The example of inducible operon is our lac operon, which I taught in the previous video, and the example of repressible operon is our trip operon, or we can say it tryptophan operon, which we will learn in today's video. So as we know, what is an operon? Operon is a segment, or we can say unit of bacterial gene expression and regulation. Suppose this is a full length DNA. It is having several genes like this: gene 1, gene 2, gene 3, so on. It will be having several genes. So now these particular genes are expressed whenever they are needed. In case of lac operon, we learned that what lac is an inducer molecule. It is an inducer molecule. Molecule means what it does? It induces the gene to switch on or switch off. Okay, so in case of lac operon, it was inducing the gene to get into on stage from the off stage. Okay, but reverse is the case in case of tryptophan operon. That is, the tryptophan operon is always in a state of on position. It is always in a state of on position by default. By default, it is always in a on position, and it continuously synthesizes the tryptophan. It continuously synthesizes the tryptophan amino acid. But what the repressible operon does, the function of repressible operon is to turn off the on position of the tryptophan operon. I hope it is clear. Now let's understand it one by one. Okay, so what is a tryptophan operon? Tryptophan operon is a repressible operon. As I told just now, tryptophan operon is a repressible operon. What it does, it encodes enzymes for synthesis of amino acid tryptophan. It encodes enzyme for what? For the synthesis of amino acid, and that amino acid is tryptophan. That is why this operon is called tryptophan operon. Now, by default, as I said just now, by default, it is always present in an switched on state. It is always present in a switched on state, but it can be repressed or it can be switched off with the co-repressor molecule, with the help of a co-repressor molecule, and co-repressor molecule in this case is our tryptophan. Okay, co-repressor molecule is in this case is our tryptophan. Means what? It is always in on position. Tryptophan operon is always in an on position, and it is turned off with the help of what? With the help of tryptophan itself. That is, this operon, trip operon, is synthesizing what? It is synthesizing tryptophan operon. This is trip operon. The function of trip operon is to synthesize tryptophan. Okay, it is synthesizing tryptophan. And this tryptophan, when present in excess, when this tryptophan is present in excess, it inhibits the trip operon. Okay, I think it is clear now. Now let's move on to next point. Now, as I told, the operon is made up of several components. Similar is the case of our trip operon. Trip operon is made up of several components. What are the components? It is made up of basically four components. What are the four components? One component is our regulator. Okay, it includes one component is our regulator, second component is our promoter, operator, and then our structural genes. Like the lac operon, it is also made up of four components: our promoter, regulator, operator, and structural genes. And in this case, structural genes are five genes. Structural genes are five genes. In case of lac operon, we studied there are three structural genes, that was lac A, Y, and Z. In this case, we have five structural genes. Okay, and these five structural genes are transcribed into a single mRNA. Now let's see the structure of lac operon. This is the structure of lac operon. Okay, for simplicity, I have drawn it like this. You can see here the first component is our regulator, second component promoter, third is operator, and the fourth component is our structural genes. Likewise, it is the structure of tripoperon. Now let's move to see this. Okay, what are the functions of different components? Just like lac operon, these have the same functions. That is, in case of regulator, what regulator does? Regulator synthesizes a regulatory or repressor protein. Regulator synthesizes a repressor protein, which is actually inactive repressor, which is inactive in nature. And what promoter does? Promoter act as the binding site for what? For RNA polymerase. It acts as the binding site for the RNA polymerase. Number three is our operator. What operator does? Operator is the binding site for the active repressor. Active repressor. This is an inactive state. Okay, when it gets activated, it will get activated when it will combine with tryptophan. Okay, when it will combine with tryptophan. Along with tryptophan, it becomes activated. And this active repressor will bind with what? With the operator region. So, operator is the binding site for the active repressor molecule. And what the structural gene does? Structural genes have certain functions, that is synthesis of the mRNA. Okay, it will first synthesize an mRNA. The five structural genes are E, D, C, B, A. Likewise, when the RNA polymerase will read this structural gene, it will synthesize the respective mRNAs, and from these respective mRNAs, there will be synthesis of the enzymes. And these enzyme subunits together will form the will help in the biosynthesis of tryptophan. Okay, I hope it is clear. Now let's move to the two cases. What happens when the tryptophan is present in the medium, and what happens when tryptophan is absent in the medium? Okay, now let's understand that. Okay, moving on to next slide. Here you can see the first case, that is when tryptophan is absent in the medium. Tryptophan is absent in the medium. As I said, when tryptophan is absent, by default, the nature of tryptophan is it is always in a on position. It is always in a on position. It will always try to synthesize what? It will always try to synthesize tryptophan amino acid. So what happens in the absence of tryptophan in the cell? This promoter region, okay, this promoter region, it has the our RNA polymerase, it and it continuously read this structural genes. Okay, and the tryptophan synthesis occurs continuously. Let's understand this. Suppose this is the case 1. In the absence of tryptophan operon, what happens? This regulator protein synthesizes the repressor molecule, but since the repressor molecule is inactive, it binds with the operator region, but it has no effects of its own. Now, what will happen? The RNA polymerase will simply pass over the structural genes without getting obstructed by the inactive repressor, and this will lead to the synthesis of mRNA transcription, and mRNA will be formed, and from mRNA translation will occur, and the enzyme subunits for the synthesis of tryptophan will easily synthesized like this. And this transcription is occurring means this is in a on state. That is, this is in a on state. That is, by default, the tryptophan operon is always in a on state, as we studied right now. And now let's understand the second case. What happens in the second case when tryptophan is high in the cells? Tryptophan is high in the cells means what? The cell is continuously, the tryptophan operon was in on condition and it was continuously synthesizing what? Tryptophan. It was continuously synthesizing tryptophan. And when tryptophan quantity is high in the cell, okay, when the tryptophan quantity is high, what will happen? It will act as a negative feedback inhibition. It will act as a negative regulator, or we can say it as a feedback inhibition. What we mean by feedback? Feedback means it will give a feedback that now this quantity of tryptophan is enough high in the cell, you must stop the tryptophan operon. What will it does? It simply goes and binds with the trypoperon, and it will inhibit the synthesis of tryptophan. It will inhibit the synthesis of tryptophan by switching it off. Okay, this is what it is written here. When the tryptophan is high in the cell, it binds with the repressor. It will bind with the repressor protein and change its conformation. The shape of the repressor will be changed, and what will happen? And it will become active. Means our repressor plus our co-repressor. Co-repressor. What is the co-repressor here? Co-repressor is our tryptophan. Okay, this will combinedly become an active repressor. Active repressor. This will combinedly form an active repressor molecule. And this active repressor molecule, what will it do? It will inhibit the tryptophan operon. So it will bind to the operator region near the promoter and it will inhibit. How it will inhibit? Binding of the repressor protein to the operator overlaps the promoter region. Okay, so the RNA polymerase cannot bind to the promoter region. Hence, transcription is halted or stopped. Okay, let's see. And this, since the excess of tryptophan is regulating the tryptophan operon, this is called a feedback inhibition, or we can say a negative regulation. Okay, let's understand with the help of this animation. Okay, so this is the regulator protein. As we know, regulator protein synthesizes the repressor molecule. Now, this repressor molecules in the presence of tryptophan will bind with the tryptophan molecule. That will result in the formation of an active repressor. Now, this active repressor will bind with the operator region and this will stop the movement of RNA polymerase like this. Okay, since the movement of RNA polymerase is stopped, hence transcription is stopped. And when transcription is stopped, there will be no more synthesis of what? Tryptophan. And since no more synthesis of tryptophan, the operon will move into an off condition. That is, to summarize, tryptophan was earlier in an on condition, now it is turned off. How it is turned off? With the help of tryptophan itself. With the help of tryptophan itself. So this is all about the tryptophan operon. We studied about how tryptophan operon acts. What is the structure of tryptophan operon? Okay. So this is all. I hope the concept of tryptophan operon is clear with this video. If you are liking this video, please drop a comment to me so that I can make more such videos. Okay. Thank you all.